Hello everyone, Dr. Ron Bio here to discuss the process of making biological polymers. These reactions in this slideshow are intentionally generic and can be used to describe the construction of macromolecules like carbohydrates, proteins, and nucleic acids, all of which are macromolecules because they can grow to be very big in size. In a dehydration reaction, you can bring together monomers through a covalent bond, resulting in the formation of a water molecule. So we're bringing these two monomers together, and basically you remove the hydroxyl group here and the hydrogen here, producing a bond, but also producing a water molecule. This process can repeat to make large polymers. In this case, we see two monosaccharides coming together to make a disaccharide, and when this repeats, you can produce your molecule all the way up to the polysaccharide level. What is not shown here is that an enzyme is required for this process. The opposite reaction is also important, as we also need to break down polymers into their constituent monomers. Hydrolysis uses a water molecule to break monomers off of the larger polymer, or in the image here, to break disaccharide into two monosaccharides. As in the dehydration example, an enzyme is also required to break this disaccharide with the help of water down into these monosaccharides. Carbohydrates are a large group of organic molecules comprised of a diverse array of structures and functions. There are many types of monomers, and these monomers can pair up in different combinations, which we will discuss at a future date. Here you see a glucose monomer and a fructose monomer being joined together to create the disaccharide sucrose. This is an example of a dehydration reaction. Again, you're removing this hydroxyl group and this hydrogen while you make this glycosidic bond. So let's apply what we have learned in an application type example. So think about this. If you have a fictitious polymer with 7 subunits, how many water molecules will you need to separate all the monomers from each other? So you have 1, 2, 3, 4, 5, 6, 7 monomers. Well, ask yourself, how many bonds are there holding those 7 monomers together? 6 hydrolysis reactions would be required to separate these monomers. So you would need 6 water molecules to break here, break here, break here, break here, break here, and break here. Okay, that's it for now. You'll need this information in your back pocket when we start to talk about how specific macromolecules are constructed in future lessons. So make sure you bring along with you this idea of dehydration reactions making polymers and hydrolysis reaction breaking polymers. As always, thank you to OpenStax for freeing the textbook and making these images available to all.